Welcome to another episode of the Med Madness podcast. Today we delve into a fundamental topic in cell biology and pathology, Integrin and Fibronectin in cellular adhesion. Understanding these molecules is crucial for medical students as they play pivotal roles in cell matrix interactions, influencing everything from wound healing to cancer metastasis. In this episode, we will cover the basics of cellular adhesion and the role of integrins, the function and importance of fibronectins, the clinical significance of Integrin-Fibronectin interactions, especially in tumour behaviour. By the end of this episode, you will have a clear understanding of how these molecules work together to maintain cellular functions and what happens when these processes go awry. Next, let's discuss cellular adhesion and integrins. Cellular adhesion is the means by which cells bind to each other's surfaces, or the extracellular matrix, also known as ECM. Integral to this process are integrins, a family of transmembrane protein receptors. Integrins interact with the ECM by binding to specific proteins, including collagen, fibronectin, and laminin. These interactions are essential for maintaining tissue structure and function. Integrins have both extracellular and intracellular domains. The extracellular domain binds to ECM components, while the intracellular domain interacts with structural proteins such as actin and keratin within the cytoplasm. This dual interaction is crucial for transmitting signals from the ECM to the cell's interior, influencing cell behavior and function. Next, let's talk about fibronectins. Fibronectins are large glycoproteins produced by fibroblasts and some epithelial cells. They serve as mediators of cell adhesion and migration by binding to integrins, matrix collagen, and glycosaminoglycans. The interaction between fibronectin and integrins is vital for cellular processes such as wound healing, embryogenesis, and immune responses. Next, let's explore the clinical significance of Integrin-Fibronectin interactions. The differential expression of integrin subtypes affects the adhesion properties of individual cells. This variation is particularly significant in the context of cancer. For example, in melanoma, changes in integrin expression can correlate with malignant behavior, influencing the tumor's ability to invade and metastasize. Consider the case of a 45-year-old patient diagnosed with melanoma. Despite initial treatment, the cancer metastasized to other organs. Upon investigation, it was found that the tumor cells exhibited altered integrin expression, facilitating their detachment from the primary tumor and invasion into distant tissues. This case highlights the importance of Integrin-Fibronectin interactions in tumor progression and underscores the potential for targeting these pathways in cancer therapy. In summary, integrin and fibronectin play critical roles in cellular adhesion and migration. Understanding their functions and interactions is essential for medical students, as these molecules are involved in numerous physiological and pathological processes, including cancer metastasis. I encourage you to engage with the pre- and post-podcast quizzes and use Anki Flashcards for effective study. These tools will reinforce your understanding and help you retain key concepts. Thank you for listening to this episode of Med Madness. Stay curious and keep learning.